welcome to the Deep Dive. Today, we're embarking on a journey that begins with just a single cell and ends with, well, you. We're talking about the truly astonishing process of animal development, how a solitary fertilized egg transforms through just spectacular complexity and precision into a fully formed, intricate multicellular organism. Our mission on this deep dive is to really unpack the fundamental cellular and molecular mechanisms that direct this amazing feat of self-assembly. We're going to be sort of pulling back the curtain on how life orchestrates itself from the ground up, hopefully giving you a fresh perspective on how we're all built.
And what's really remarkable, as we've seen in the sources we're looking at, is that while, plants and animals use different strategies, much of the core machinery, the tools of development, are conserved across all animals. Think about it. A tiny worm and, us, complex humans, we share a surprising number of the same molecular tools for building a body.
Wow.
We'll primarily focus on animal development today, which should give us a pretty comprehensive view of this field.
I find that absolutely mind-boggling, just the sheer efficiency of evolution. So we're going to trace this incredible journey, how cells multiply from 1 to billions, how they specialize, differentiate, how they arrange themselves into precise tissues and organs, and even how an organism's final size is ultimately determined. Get ready for some truly illuminating moments, I think, that connect the tiniest molecular details to the grand scale of life itself. Let's dive in. An overview of development, from zygote to specialized forms. All right, let's zoom in on the opening acts of this biological play. How does a single cell even begin to build something as complex as us? It seems to boil down to 3 fundamental processes, like the master choreography of development. First, there's cell proliferation. This is simply how one cell becomes many, through repeated division. Pretty straightforward. Right. Then there's cell specialization, or differentiation. That's how those cells take on different characteristics and functions, becoming, say, a skin cell, or a neuron, or a muscle cell, each with a unique job. And finally, morphogenesis. This is where cells literally rearrange themselves, pushing and pulling, to form structured tissues and organs, creating the very architecture of the body. It's like the ultimate biological self-assembly kit in action.
That's a great way to put it. And those initial stages, they're incredibly rapid. That single fertilized egg, the zygote, immediately begins what's called the cleavage phase.
Cleavage, okay.
Yeah, this is a period of fast, repeated cell divisions, creating many smaller cells, which we call blastomeres. What's intriguing here is that the embryo doesn't actually grow in overall size during this stage.
Really? So it's just dividing?
Just dividing, getting smaller cells. And it's initially driven and controlled entirely by the maternal material, special mRNAs and proteins that were pre-packed into the egg by the mother. The embryo's own genes aren't active yet. Then comes a crucial turning point, often called the maternal zygotic transition. This is when the embryo's own genome finally activates and takes over control.
Oh, okay. It wakes up.
Exactly. The cells then cohere or stick together to form a blastula. You can picture this as a simple, maybe hollow ball of cells. What follows is the absolutely crucial process of gastrulation. This is a complex series of coordinated cell movements and rearrangements that transform that simple blastula.
Right.
Cells fold inwards, they push and pull, turning that sphere into a more complex, multi-layered structure, often with the beginnings of a rudimentary gut already forming inside. It's a major reorganization.
And those layers are so fundamental, they're truly a blueprint. Gastrulation gives rise to the 3 primary germ layers, which are remarkably conserved across pretty much all animals and define the basic tissue types. We have the ectoderm, which stays on the outside and will form things like your epidermis, your outer skin, and your entire nervous system, including your brain and spinal cord.
Right, the outer stuff.
Then the endoderm moves inwards to form the gut tube and its associated organs, like your lungs, pancreas, and liver, the inside lining. And finally, sandwiched in between is the mesoderm. This will form your muscles, all your connective tissues, blood, kidneys, and even the notochord, which is the precursor to your backbone in vertebrates.
That's the middle layer. Yeah, it forms a lot of the bulk.
These 3 layers are the foundational building blocks for everything else.
And what's fascinating here is how the developmental potential of these cells becomes progressively restricted. Early on, cells in that blastula stage are often described as totipotent or pluripotent.
Meaning they can become anything.
Almost anything, yeah. They have the potential to form all or nearly all cell types of the adult body. But as gastrulation proceeds, their fate becomes determined, narrowing their options. An endodermal cell, for instance, can form gut lining cells or pancreatic cells, but it won't suddenly decide to become a muscle cell. Its choices are limited.
like closing doors on potential career paths.
Kind of. It's a step-by-step process towards specialization until cells undergo what we call terminal differentiation, becoming a highly specialized adult cell type, like a neuron or a red blood cell. This leads us to a really important concept: cell memory. Cells maintain their specialized characteristics, like being a skin cell versus a muscle cell, not because they're constantly receiving external instruction.
They don't need constant reminders.
No, they retain a record of past signals. Their identity becomes stable. This memory is largely due to differential gene expression. Different sets of genes are stably turned on or off, defining their unique character. Once a cell commits, it remembers.
So if these mechanisms are so fundamental and often happen on a microscopic scale, how do scientists even begin to untangle such complexity? You mentioned sources rely on model organisms.
That's a great question. We really owe much of our understanding to the power of model organisms. Scientists have uncovered many of these universal mechanisms by studying species like the fruit fly Drosophila melanogaster.
The classic.
The classic, yeah. Also the frog Xenopus laevis, the tiny worm Canarhabditis elegans, or C. elegans the mouse, and the zebrafish. What's truly striking is how conserved many of these proteins and their functions are across these vastly different animals. Take, for example, the eyeless protein in Drosophila. It controls eye development. Amazingly, if you take the homologous protein from, say, a mouse that's called PAX6 and introduce it into a developing fly, it can still induce eye tissue formation, sometimes in really weird places, like on a fly's leg or wing.
Wow, so the mouse eye-building protein works in a fly.
It triggers the fly's own eye-building program. It's an incredible demonstration of this shared molecular toolkit across species, and that leads to a truly profound insight into evolution. The vast differences between, say, a fly and a human aren't primarily because we have entirely different sets of basic building block proteins.
Oh.
No, evolution is incredibly resourceful. It's largely achieved this staggering diversity by taking the same set of molecular tools, those conserved proteins, and simply writing different instruction manuals for how and when to use them.
Changing the instructions, not the parts.
Exactly. These new instructions are written in the regulatory DNA, the parts of our DNA that control when and where genes are turned on or off, not the coding sequences of the proteins themselves. Think of it, same Lego bricks, but building wildly different things just by changing the blueprint.
That's a powerful way to put it. And how do cells get those intricate instructions for assembly? You mentioned signals.
Right. It's largely through cell signaling. This is absolutely crucial for patterning an embryo. One common method is inductive signaling.
Okay.
Here, a signal from one group of cells can quite literally induce or instruct nearby cells to change their character. It's like a biological instruction manual being passed directly from cell to cell, telling them what to become.
What I find truly remarkable about this is the efficiency. It seems like the body, despite its vast complexity, relies on a surprisingly small set of key signaling pathways, reusing them in different contexts.
Yep.
How do these few pathways manage to generate so much diversity?
That's a brilliant observation. You're absolutely right. There's a surprisingly small number of highly conserved signaling pathways like TGF, WANT, hedgehog, notch, and receptor tyrosine kinase pathways that are used over and over again throughout development.
So how does that work?
The diversity comes from two main principles: combinatorial control and cell memory. A cell's response to a signal doesn't just depend on that single signal alone, it also depends on what other signals it's currently receiving.
The combination matters.
Precisely. And crucially, it depends on its unique history, its cell memory, which influences how it interprets those signals. So a cell that has previously been exposed to one set of signals will respond differently to a new signal than a cell with a different past.
Okay, so context is everything.
Context and history, exactly. It's like different instruments in an orchestra, each playing the same note but producing a unique sound depending on its tuning and what else is playing. Building on that, morphogens are a key concept in how these signals create patterns. Imagine a tiny biological smoke signal spreading out from a source, creating a concentration gradient.
Like ripples in a pond.
Sort of, yeah. These are diffusible inductive signals. Cells respond differently based on how much of that morphogen signal they receive. A high concentration might direct cells down one developmental path, while an intermediate or low concentration pushes them down another.
So position determines fate based on signal strength.
Exactly. It's a precise way to tell cells their position along an axis. And the speed at which the signal diffuses and how long it lasts its half-life determine the range and steepness of this gradient, ultimately shaping how clearly distinct cell fates are established. Another remarkable mechanism is lateral inhibition. This is a powerful form of competition where cells exchange signals, forcing close neighbors to become different from each other.
Competition, how does that work?
Imagine a group of similar cells, all with the potential to become, say, a neuron. If one cell starts making a little more of a signal that inhibits its neighbors,
It tells them back off.
pretty much, it amplifies that initial difference, making itself the specialized cell, while forcing its neighbors into a different, maybe less specialized role. This often involves the notch signaling pathway and is essential for generating fine-grained, evenly spaced patterns, like the bristles on a fly's back.
But not all diversification depends on external signals from other cells, right? You mentioned cells can be born different.
Exactly. Sometimes daughter cells are actually born different as a result of asymmetric cell division. In these cases, key molecules inside the mother cell are unequally distributed between the two daughter cells during mitosis.
So they inherit different stuff.
Precisely. This ensures that even without any new external instructions, one daughter cell inherits specific molecules that direct it down one path, while the other daughter, inheriting different molecules, takes a different path. It's a pre-programmed way to generate diversity right from the start. And initial patterns, often established when the embryo is small, get refined through what we call sequential induction as the embryo grows.
Sequential induction, meaning one step leads to the next.
Exactly. A simple initial pattern can be elaborated with increasing levels of detail through a series of successive inductive interactions. For example, one cell type induces a second, which then signals back, maybe creating a third, and so on. It's like a chain reaction, progressively building complexity and adding more specific details as development proceeds.
This might seem like highly abstract biology, but the progress in understanding animal development has profound practical implications, doesn't it?
Oh, absolutely. For one, it provides crucial insights into how birth defects arise, which unfortunately affect a significant percentage of human babies.
Right.
But even more broadly, these developmental processes don't just stop at birth. They continue throughout our lives. They're essential for adult tissue maintenance and repair. Think about how your skin heals or how blood cells are constantly replaced.
So understanding development helps understand adult life, too.
Definitely. And understanding these fundamental mechanisms of cell growth, signaling, and movement is also crucial for understanding diseases like cancer, because those are the very processes, proliferation, migration, differentiation, that go awry in tumor formation and progression. 2. Mechanisms of pattern formation. Establishing the body plan.
Okay, now let's dive deeper into how those exquisite spatial patterns of specialized cells are generated. Basically, establishing the animal's entire body plan, mapping out where the head, tail, back, and belly will actually be.
Right. This mapping begins with establishing the primary body axis. We have the anteroposterior AP axis, which specifies head versus tail.
Steps back.
Exactly. Then the dorsiventral DV axis for back versus belly.
Top to bottom, essentially.
And for some species, a left-right LR axis, which dictates things like heart placement. Many animals also have an animal-vegetal AV axis that defines which parts become internal during development. What's surprising is the variability in how these axes are initially set up across different species. A mouse egg, for instance, shows very little initial polarity, but others, like Drosophila or the frog Xenopus, have very clear, predefined axes right from the start in the egg.
So different strategies to get started.
Definitely. Take the Xenopus frog embryo as a classic example. Its AV axis is already defined before fertilization, with specific maternal mRNAs, like VegT mRNA, localized at what will become the vegetal pole, the bottom. But here's the really cool part. Fertilization itself is the trigger for the DV axis.
Hope so.
The sperm entry point, wherever it happens to hit, initiates something called cortical rotation. The outer skin, or cortex of the egg, slowly shifts relative to its inner contents. This subtle but profound reorganization moves signaling molecules around and establishes the future dorsal side opposite the sperm entry point.
Wow, so sperm entry breaks the symmetry.
It's a beautiful example of symmetry breaking, transforming a seemingly uniform ball into something with clear directional information.
While frogs are fascinating, you mentioned the power of Drosophila genetics. How did flies help us understand this? They have that weird early stage, right?
They do. Drosophila development has this peculiarity. After fertilization, the nuclei divide repeatedly without the cell actually dividing. It forms a syncytium, essentially a giant cell bag with many nuclei inside.
Like one big room with lots of workstations.
Exactly. These nuclei then migrate to the periphery to form the syncytial blastoderm. Only later does the plasma membrane fold inward to partition them into separate cells, creating the cellular blastoderm. This unique early syncytial stage is critical because it allows transcription regulators, proteins that turn genes on or off, to diffuse relatively freely through the cytoplasm and act directly on many nuclei at once without needing cell signaling initially. That's a major difference from vertebrates like frogs.
Like, okay, so things can spread easily in that early stage.
Right. And within this environment, egg polarity genes in Drosophila are absolutely critical. These are maternal effect genes. Their products, mRNA or protein, are deposited by the mother into the oocyte before fertilization. They act like molecular landmarks defining the main axis.
So the mom sets up the initial map.
Precisely. A famous example is bicoid. Its mRNA is specifically localized at the anterior, the future head end, of the egg. After fertilization, it gets translated into protein. The bicoid protein then diffuses away from the head end, forming a concentration gradient highest at the head, decreasing towards the tail. This gradient acts as an intracellular morphogen and transcription regulator.
So the amount of bicoid tells the nucleus where it is.
Essentially, yes.
Oh.
It directly regulates gene expression in the nuclei based on their position along that gradient, thereby determining the formation of head structures. Other egg polarity systems, like nanos, toll, and torso, work in similar ways using gradients or localized activation to establish other crucial body partitions.
From these initial broad gradients, Drosophila development then moves into segmentation, subdividing the AP axis into those repeating segments we see in flies.
Right, and this involves a hierarchy of genes expressed by the embryo's own genome, now the zygotic effect gene.
The cascade of instructions.
Exactly. First, gap genes, like Krupel, mark out coarse AP subdivisions. A mutation here can cause the embryo to lose entire groups of segments, leaving big gaps. Next, pair rule genes refine this pattern. They cause deletions affecting alternate segments, so an embryo might end up with only half the usual number of segments in a striped pattern.
Odd.
Very odd. And finally, segment polarity genes define the internal pattern of each individual segment. A mutation in a gene like hedgehog here can cause mirror image duplications within segments. So it builds a sophisticated regulatory hierarchy. Egg polarity genes give the global position, gap genes refine into broad domains, pair rule genes demarcate the segments, and segment polarity genes pattern within each segment.
It's intricate.
It is, and those segment polarity genes, like wingless, part of the want pathway, and hedgehog, are synthesized in different bands of cells. They actually signal to each other, mutually maintaining their own expression, while also regulating other genes like engrailed in neighboring cells.
A feedback loop.
A crucial feedback loop. This continuous signaling creates stable boundaries and patterns within each segment, with genes like engrailed marking boundaries that persist throughout the fly's life.
Now we come to one of the most remarkable discoveries in developmental biology, Hox genes. These are master regulators that permanently pattern the AP axis, giving cells their address along the body.
Ah, yes, the homeotic genes.
Right. The first clues came decades ago with these bizarre homeotic mutations in Drosophila. You mentioned antennapedia, where legs sprout from the fly's head instead of antenna.
Right, completely wild.
And bithorax, where you might find parts of an extra pair of wings instead of little balancing organs called halters. These mutations literally transform one body part into another, revealing this profound control system.
There are 8 Hox genes in the fly, organized into 2 clusters on the chromosome. These genes and their protein products are transcription regulators. They have a highly conserved DNA-binding region called the homeodomain.
And they act like address labels.
Exactly. We call these Hox proteins molecular address labels, giving cells a positional value that dictates their segment identity. In fact, if you delete all Hox genes in a fly, all its body segments end up looking pretty much alike. They lose their distinct identities.
That shows how crucial they are.
Absolutely. And what's truly astonishing about Hox genes is a principle called collinearity.
Collinearity?
Yeah, imagine the Hox genes lined up in order on the chromosome. Now picture the embryo's body axis from head to tail. The order of the genes on that chromosome matches the order of where they are expressed along the embryo's body.
No way. The gene order matches the body order.
It does. The gene at one end of the cluster controls head structures, and genes further down the line control progressively more posterior structures towards the tail. It's a striking molecular roadmap conserved across many animals.
How is that pattern even maintained as cells divide over and over?
Great question. It's through the action of 2 large complementary sets of proteins, the trithorax group and the polycomb group.
Trithorax and polycomb.
Right. They work to modify the chromatin, the packaging of DNA, creating a heritable record, a cellular memory, of whether a Hox gene should be active or repressed in that particular cell lineage. Trithorax proteins generally maintain gene activation, while polycomb proteins form stable complexes that keep genes silenced. If these groups are defective, the Hox pattern isn't maintained correctly, leading to severe developmental problems. They're fundamental for that long-term cell memory.
Okay. Shifting from AP to the DV axis in Drosophila, the back-to-belly patterning, that also starts with maternal signals.
It does. A protein signal from the mother's somatic cells activates a receptor called Toll, but only on the ventral belly side of the egg membrane. This localized activation controls where a key transcription regulator called dorsal ends up. In that early syncytial blastoderm, dorsal protein gets highly concentrated in the nuclei on the ventral side, with a smooth gradient decreasing towards the dorsal backside.
Another gradient.
Another gradient, yes. Like bicoid for the AP axis, dorsal acts as a morphogen for the DV axis. Its concentration inside the nucleus turns different genes on or off, defining distinct DV territories.
So high dorsal means one thing, low dorsal means another.
Exactly. The highest dorsal concentration ventrally switches on the twist gene, crucial for making mesoderm. Most dorsally, where dorsal is lowest, cells activate the decapentaplegic DPT gene, and in an intermediate region, the short gastrulation SOG gene is activated. Then, these initial DPP and SOG proteins, which are secreted signals, create opposing extracellular gradients. SOG actually blocks DP signaling. This competition further refines the DV territories, creating a very precise layered pattern.
Opposing gradients. That sounds complex.
It is, but it's a powerful way to sharpen boundaries. And this strategy is strikingly similar to what we see in vertebrates, showing remarkable conservation.
And that brings us neatly to vertebrate development. You keep mentioning conservation. How similar is it, really?
There's an astonishing degree of conservation in the underlying logic of pattern formation, even if the very earliest steps like axis specification might vary a bit.
Like that frog organizer experiment.
Exactly. The frog organizer is a classic discovery from the early 20th century. Researchers took a small cluster of dorsal cells from one frog embryo and transplanted it into a different region of a host embryo. The incredible result was that this tiny piece of tissue could induce a whole second body axis to form in the host a second head and trunk.
Wow. A signaling center.
A powerful signaling center, yes. It revealed this chain of inductive interactions that builds the vertebrate body. And unlike the Drosophila syncytium, frog embryos divide into distinct cells early on, so patterning relies heavily on extracellular signal molecules diffusing between cells. That organizer region is a major source of these secreted signals.
So what signals are involved?
Well, along the AV axis in frogs, you have growth factors from the TGF family called nodal, and its antagonist lefty forming opposing gradients. Nodal induces endoderm and mesoderm, while lefty blocks nodal further up, leading to ectoderm. For the DV axis, it's a competition between bone morphogenetic proteins, BMPs, and their antagonists like chordin and noggin, which are secreted by the dorsal signaling system, the organizer.
BMPs. Like the DBP in fly.
Very much analogous, yes.
Yeah.
This creates a DV-BMP gradient. High BMP leads to epidermis, skin, while low BMP allows neural tissue, brain, spinal cord, to form. It's deeply homologous to the DPP SOC system in flies. The names are different, but the logic is the same.
And the Hox genes, are they conserved too?
Absolutely. Vertebrate Hox genes are highly conserved. Mammals, including us, typically have 4 Hox complexes, Hox A, B, C, and D, each on a different chromosome.
4 sets.
4 sets, likely due to whole genome duplications in vertebrate evolution. Individual genes in these complexes are direct molecular counterparts of the Drosophila Hox genes, and they even show the same remarkable collinearity.
Still matching the body axis?
Still matching. Their order on the chromosome matches their expression along the AP axis of the embryo, controlling patterning in regions like the hindbrain, neck, and trunk. And just like in flies, gain or loss of these genes can literally transform tissue identity, like changing one type of vertebra into another.
It's not just regional identity, though, right? Some genes control whole cell types or organs.
Exactly. Certain master transcription regulators can act like a switch to trigger the development of specific cell types or even entire complex organs.
Like what?
For cell types, think of MyoD, which can turn many cell types into muscle, or a K-discute, which prompts cells to become neural progenitors. For organ formation, the transcription regulator ILUS, or PAX6 in vertebrates, is a prime example.
The I gene again?
The I gene. Mutations in PAX6 lead to a complete lack of eye structures in both flies and humans, showing its fundamental conserved role. However, these powerful regulators still depend on the context and history of the cells they act upon. They don't just magic an eye out of nowhere. They flip the switch in cells that are already competent or prepared to become eye tissue.
Okay, so after the basic body plan is set, there's still refinement needed. You mentioned notch-mediated lateral inhibition earlier for fine-grained patterning.
Right. It's crucial for creating patterns where specialized cells are interspersed among non-specialized cells in a regular way. Think about the sensory bristles on a Drosophila fly's back. They're very evenly spaced.
How does Notch do that?
Within a cluster of cells that all initially have the potential to become a bristle, they start competing via notch signaling. If one cell gains a slight advantage, it sends strong inhibitory signals to its immediate neighbors through the notch pathway.
Telling them you can't be a bristle.
Exactly. This prevents them from doing the same, ensuring that only one cell becomes the sensory organ precursor in that little neighborhood, creating that precise, spaced-out pattern. It's a fundamental mechanism for generating regular arrays of distinct cell types.
And we also talked about asymmetric cell division as another way to get diversity without external signals.
Yes, another powerful internal mechanism. For example, in frog embryos, that veg-tRNA we mentioned is asymmetrically localized in the egg, so only certain cells inherit it after division, setting up early differences. Similarly, in Drosophila bristle development, a protein called NUM gets segregated to only one side of the cell before it divides. The daughter cell that inherits NUM takes one fate, like becoming a neuron, while the daughter cell without NUM takes another, like becoming a supporting socket cell.
So the cell itself distributes key molecules unevenly.
Precisely. This differential inheritance directly dictates distinct cell fates from a single mother cell, all pre-programmed.
All these molecular insights, they really circle back to the incredible story of evolution, don't they? Especially the idea about regulatory DNA.
Absolutely. It's becoming clearer and clearer that many of the profound morphological differences between species, the visible changes in body shape and structure, are largely accounted for by changes in regulatory DNA rather than just changes in the protein coding sequences themselves.
Can you give an example?
A really compelling one is the stickleback fish. Marine sticklebacks have prominent pelvic spines for defense, but many freshwater populations have lost these spines entirely.
Why?
It turns out it's often not due to a change in the PITSLX1 protein itself, which is involved in hind limb pelvic development. Instead, it's frequently caused by a small deletion in a specific regulatory DNA element, an enhancer that normally drives PITSLX1 expression only in the developing pelvic region.
So the gene is fine, just the on switch for the pelvis is broken.
Exactly. The gene is still expressed elsewhere in the body where it's needed, but the instruction to build the spine in that specific body part is simply removed. This leads to the loss of the structure. It's a beautiful demonstration of how evolution can subtly tweak developmental instructions to create significant diversity.
That makes sense.
And the evolution of maize from its wild ancestor Teosinte is another great example. Dramatic morphological differences between the spindly wild grass and the robust corn cob we know today were driven by relatively few changes, many in regulatory regions of key developmental genes, sometimes even involving insertions of mobile genetic elements that altered gene expression patterns. 3. Developmental timing. The orchestration of ants.
Okay, let's transition to, well, an equally fascinating, but maybe, as you said, least understood aspect of development, timing. How do all these incredibly complex events unfold over precise minutes, hours, or even years? What sets the biological clock?
Yeah, timing is critical, and it's complex. One fundamental factor is simply molecular lifetimes. It's not just how quickly cells synthesize new molecules like mRNA or proteins.
There's a delay.
There's an inherent delay. It takes time for mRNA to be processed, exported from the nucleus, translated into protein. And then those proteins need time to accumulate to an effective concentration to actually do something. These gestation and accumulation times, which are largely determined by how long the mRNA and protein molecules persist before being degraded their half-lives, they dictate the delays in gene-switching events.
So the stability of molecules matters for the tempo.
Absolutely. Cascades of gene activation and signaling pathways accumulate these delays, and that collectively controls the precise pace and tempo of development.
One of the most striking examples of a biological clock in action must be the vertebrate segmentation clock. Our bodies, and those of all vertebrates, have this repetitive structure. Vertebrae, ribs, muscles, all forming repeating units.
The somites.
Right, the somites. They originate from these segments that form sequentially, one after another, in a remarkably regular rhythm, unlike Drosophila segments, which seem to appear all at once.
Right, simultaneously.
Vertebrate somites bud off in this precise clock-like fashion, starting near the head and moving towards the tail. It's like a biological assembly line, adding new segments at a fixed tempo.
It really is. The cells that form these somites come from a region at the posterior end called the presemitic mesoderm. And here, the expression of certain genes, particularly the Hes genes, oscillates in time, like a molecular pendulum.
Hes genes?
Yes, they encode inhibitory transcription regulators that actually block their own expression. So as Hes protein builds up, it shuts off its own gene. Then as the protein decays, the gene turns back on.
A negative feedback loop.
Exactly, a cyclical negative feedback loop. This internal oscillator acts as the pacemaker for somerite formation. The period of this oscillation, how fast the clock ticks, is determined by the total delay in that feedback loop, essentially the molecular lifetimes of the Hes mRNA and protein.
So each cell has its own clock.
Each cell can oscillate on its own. But what's really cool is that another signaling pathway, Notch, acts between neighboring cells to synchronize these oscillations.
Keeps them all ticking together.
Precisely. It prevents chaotic segmentation. If Notch signaling fails, the cells drift out of sync, and you get gross deformities where the segments are completely disorganized. Synchronization is key.
It's incredible to think about these internal molecular pacemakers. But not all developmental changes require external signals or these synchronized clocks, right? Some cells seem to have cell intrinsic timing mechanisms.
That's right. Some cells seem to run on their own internal schedule. For instance, Drosophila neuroblasts, the stem cells of the fly's central nervous system, undergo a fixed sequence of asymmetric cell divisions. As they divide, they sequentially express different transcription regulators, proteins like hunchback, cripple, PDM, and castor, in a precise pre-programmed order over time.
Changing identity with each division.
Well, changing the type of neuron or glial cell they produce with subsequent divisions. This allows one stem cell to generate a whole diversity of distinct cell types at fixed times during development. And this internal program continues even if the cells are isolated in culture or if cell division itself is blocked.
So it's not just counting divisions.
Exactly. It suggests it's not the cell cycle itself that acts as the timer, but rather the internal dynamics of these developmental control genes changing over time. We see similar processes in the developing mammalian cerebral cortex, where the birth order of neurons strongly influences their final fate and their position in the layered structure of the cortex. And that's a crucial point. It's a common misconception that cells count their divisions to time development. While many specialized cells do stop dividing after a limited number of divisions, experiments blocking cell division show that cells can often still mature and differentiate on schedule.
So the internal gene program is the timer.
It seems the developmental control genes, not just the cell cycle clock, really set the tempo and dictate when certain events occur.
Another surprising discovery in developmental timing came from those genetic screens in the nematode worm C. elegans. That tiny worm with the perfectly mapped cell lineage.
Right, its development is incredibly precise and predictable. Scientists know the ancestry of every single adult cell.
And these screens found heterochronic mutants. What were those?
They were mutants where developmental timing was disrupted. You'd see cells behaving as if they were stuck in an earlier larval stage, or maybe skipping a stage entirely, messing up the normal sequence.
And what caused that?
What they found was truly groundbreaking. Two key genes involved in these timing pathways, called Lin4 and Lit7, didn't code for proteins at all. They coded for tiny RNA molecules called microRNAs.
Rearrangements.
MicroRNAs, tiny RNAs controlling timing.
Exactly. These short regulatory RNA molecules bind to complementary sequences in the messenger RNAs of other heterochronic genes, like Lin14 or Lin41. This binding acts like a dimmer switch, repressing the translation of those target mRNAs into protein and often promoting their degradation.
So they fine-tune the levels of key timing proteins.
Precisely. For example, the Lin4 mirin helps trigger the progression from an early to a late larval stage by precisely reducing the levels of the Lin14 protein, which normally keeps cells in the early larval state. Lin4 and Lit7 were actually the first miRNAs discovered in animals, revealing this whole new layer of gene regulation essential for sharpening developmental transitions and keeping the developmental clock running accurately.
And speaking of transitions, how is the timing of that huge switch, the maternal zygotic transition, MZT, controlled? when the embryo's own genome finally takes over.
That's another fascinating timing question. One key trigger appears to be something called the nuclear to cytoplasmic ratio. Remember how early embryos divide rapidly without growing?
Yeah, lots of nuclei in the same volume.
Exactly. The number of nuclei, and thus the total amount of DNA, increases dramatically within a constant cytoplasmic volume. Now, imagine there's a fixed amount of some kind of maternal repressor molecule floating in that cytoplasm. Each new nucleus, with its DNA, acts like a sponge, binding up a little bit of that repressor. Eventually, there are so many nuclei that the repressor gets effectively titrated or diluted out below a critical threshold.
So the increasing DNA soaks up the off switch.
That seems to be a major part of it. This sudden drop in free repressor concentration then allows the zygotic genome to become active, flipping the switch for the embryo's own genes to finally take control of development. Beyond these localized or early timers, hormonal signals play a vital role in global coordination, orchestrating developmental events across the entire body, often over longer timescales.
Like metamorphosis.
Exactly. The dramatic metamorphosis of an amphibian from a tadpole to a frog is a spectacular example.
Yeah.
This complete bodily transformation, losing gills and tails, growing legs and lungs, is triggered and coordinated by thyroid hormone.
One hormone triggers all that.
It orchestrates it. The hormone circulates throughout the body, and different tissues respond at the right time and in the right way, leading to this massive coordinated change. And often, the release of the hormone itself is influenced by environmental cues like temperature or food supply, linking development to the outside world.
And environmental cues seem even more critical in plants, especially for long-term timing, like flowering.
Absolutely. Plants are masters of long-term environmental timing. Take vernalization, the requirement for prolonged cold exposure before many plants can flower in the spring.
How do they remember the cold?
It involves stable changes in chromatin structure, how their DNA is packaged. Prolonged cold induces these changes, often involving specific non-coding RNAs like coupled and polycomb group proteins, similar to the ones that maintain Hox gene expression. These chromatin changes effectively silence a key gene called FLC, which normally acts as a repressor, preventing flowering. By silencing FLC, the cold period essentially removes the brakes on flowering.
So it's a chromatin timer set by winter.
Pretty much. This ensures the plant only flowers after winter has passed, even when temperatures rise in spring. It prevents them from flowering too early and getting caught by a late frost. It's a fascinating molecular memory of environmental conditions.
Sculpting form.
All right, that brings us to the really physical aspects of development. How cells actually move, bend, stretch, and deform to create specific shapes and sizes. This is the art of form generation morphogenesis, where biology meets architecture and engineering.
Exactly. Morphogenesis fundamentally involves an imbalance of physical forces acting on cells. These forces can be generated internally by the cell's own cytoskeleton, particularly actin and myosin, which act like tiny muscles.
Pulling and squeezing.
Right. Or the forces can be external, coming from neighboring cells or the surrounding environment. And cells need to sense these forces. They do this using specialized proteins called mechanotransducers. Think of proteins like alpha-catenin at cell junctions, talon connecting to the matrix, or channels like piezo.
They're like tiny sensors.
Exactly. They detect mechanical stimuli, stretch, compression, shear stress, and convert that physical force into intracellular signaling. This allows cells to read their physical environment and respond appropriately, maybe by changing their shape, their adhesion, or their movement.
You can see these forces playing out even in simple structures, like how cells pack together in sheets.
Absolutely. Tension and adhesion determine cell packing in epithelial sheets. If you look at the surface of many epithelia, like your skin or gut lining, the cells often have a remarkably consistent hexagonal profile.
Like a honeycomb.
Precisely. This arrangement is a beautiful balance. You have cortical tension, generated by actin-myosin rings just under the cell surface, which tends to minimize surface area and pull cells apart slightly. And you have cell-cell adhesion, primarily mediated by cadherin proteins, which act like molecular vis zero, holding them together.
So tension versus sticking.
It's a balance, exactly like the physics of soap bubbles, where surface tension dictates their elegant hexagonal packing. And changing patterns of cell adhesion molecules, particularly those cadherins, are crucial for dynamic rearrangements during morphogenesis. Cells can actually change which specific cadherins they express on their surface.
Why does it matter?
Because different cadherins prefer to stick to themselves, like sticks to like. So if a group of cells starts expressing a different cadherin from its neighbors, they will tend to sort out, cohering with each other, while separating from the cells with the original cadherin.
Ah, creating boundaries.
Exactly. This selective stickiness is vital during processes like gastrulation, where cells sort themselves into distinct germ layers, and during neural tube formation, where the sheet of future neural cells detaches from the overlying ectoderm and folds up.
But cells don't just stick together. Sometimes they need to actively push each other away to maintain boundaries, right?
That's right. Repulsive interactions are also important. These are often mediated by a signaling system involving effrin ligands and their F receptors on neighboring cells.
Afferents and Fs.
Yeah. When an F receptor on one cell binds to an effrin on an adjacent cell, it often triggers a repulsive response, preventing the cells from mixing. This helps maintain sharp, clean boundaries between different tissue territories. We see this keeping segments separate in the developing vertebrate brain, rhombomeres, and also contributing to semimite boundaries. It's like molecular keep-out signs. And then there are large-scale movements involving collective rearrangements of similar cells. Convergent extension is a classic and really important example.
Convergent extension. Sounds like stretching.
It is. Imagine a sheet or group of cells that needs to get longer and narrower. The cells rearrange themselves, moving towards a central line, converging, while simultaneously pushing past each other to lengthen the tissue along a perpendicular axis, extending.
Yeah.
It's like a crowd of people shuffling sideways to squeeze through a narrow doorway, making the crowd longer and thinner.
How do cells do that?
It can happen in a couple of ways. In some cases, like early frog gastrulation, it involves mesenchymal cells, loosely connected cells, actively crawling over each other and intercalating. In other cases, like in Drosophila germ band extension, cells within a tightly connected epithelial sheet rearrange their neighbors by actively remodeling their junctions, breaking old connections and making new ones, all while maintaining the integrity of the sheet.
Speaking of epithelia, there's also planar cell polarity. What's that about?
So epithelial cells usually have a clear apical and bottom basal polarity. But planar cell polarity, or PCP, refers to their coordinated orientation within the plane of the tissue sheet.
It's like all facing the same way sideways.
Exactly. Think of the hairs on a fly wing. They all point uniformly towards the tip of the wing. Or the sensory hair cells in your inner ear, which need to be precisely oriented to detect sound vibrations correctly.
How is that set up?
It involves a specialized set of conserved signaling proteins, including components related to the Wnt pathway and unique transmembrane proteins like frizzled and Van Gogh, as well as specialized cadherins. These molecules become asymmetrically localized within each cell and communicate between neighbors to align the polarity across the whole tissue. If this system fails, you get defects like misoriented hairs or, in vertebrates, deafness due to disorganized inner ear cells. A really dramatic example of morphogenesis is how an epithelium can bend to form a tube. This is fundamental to forming things like our gut, blood vessels, and nervous system.
How does a flat sheet become a tube?
A common mechanism is apical constriction. Cells within the sheet, often along a specific line, contract actin and myosin filaments located just under their apical top surface. This contraction squeezes the apical side, making the cells wedge-shaped.
Like pulling a drawstring.
Exactly like pulling a drawstring. Because the cells are connected to their neighbors, this coordinated constriction causes the entire sheet to buckle inwards and eventually roll up into a tube. This is precisely how the neural tube, the precursor to our brain and spinal cord, forms.
It's pretty neat. Are there other ways to make tubes?
Yes, there are other mechanisms too, like cells rearranging or hollowing out a solid cord of cells, but apical constriction is a really common and visually striking one.
And it gets even more intricate with things like branching tubular structures. Organs like our lungs or kidneys form these incredibly complex, tree-like branching networks. How does that happen?
That process is called branching morphogenesis. It typically depends on continuous back-and-forth signaling between the growing epithelial buds, the tips of the branches, and the surrounding mesenchyme, the supportive connective tissue.
A dialogue between tissues.
Constant dialogue. For example, in lung development, the mesenchyme near a bud tip often secretes a growth factor called FGF10. This acts like an invitation, telling the epithelial cells grow out this way. But then, the epithelial cells at the very tip of the growing bud start producing a signal, often sonic hedgehog, shh, which signals back to the mesenchyme right underneath the tip. This shush signal tells the mesenchyme to stop making FGF10 locally, but encourages FGF10 production just to the sides.
Ah, so it splits the signal.
Exactly. It splits the FGF10 signal into two hotspots on either side of the original tip. This in turn causes the epithelial bud to bifurcate and grow out into two new branches, repeating the process over and over to generate that complex tree.
That's elegant.
It is. And we see similar principles, often involving related signaling molecules like FGF, driving the branching of the insect tracheal system, which delivers oxygen. Interestingly, tracheal branching can also be stimulated by low oxygen conditions via hypoxia-inducible factors, HIFs, showing how physiological needs can directly influence morphogenesis. We should also mention the extracellular matrix, ECM, that network of proteins and polysaccharides outside cells. It's not just passive scaffolding.
How does it influence shape?
It provides tracks and substrates for cell migration. Its composition and stiffness can guide cells. And the matrix itself can exert mechanical forces or resist forces generated by cells influencing tissue shape. For instance, localized breakdown or softening of the ECM can allow epithelial buds to push outwards more easily, contributing to branching patterns in organs like the salivary gland.
Beyond shaping sheets and tubes, many cells need to actively move around during development, sometimes traveling huge distances. How is cell migration guided?
Migrant cells, like the precursors to our muscles, myoblasts, or the incredibly versatile neural crest cells, are guided by a remarkable array of environmental cues.
Like following a map.
Very much like following a map laid out in the environment. This map can include pathways marked by adhesive molecules in the ECM, like fibronectin, which acts like sticky footholds. It can also include keep-out signals from repellent molecules, like certain proteoglycans. Cells are also guided by specific chemical signals, chemoattractants or chemorepellents. A well-studied example is the chemokine CXCL12 and its receptor CXCR4. Cells expressing CXCR4 will migrate towards sources of CXCL12. This guides critical migrations, like getting germ cells to the developing gonads.
So they follow the scent?
In a molecular sense, yes. But just finding the right place isn't always enough. The final distribution of migrant cells often depends on whether they survive the journey and arrival. Specific destinations provide crucial survival factors. Take neural crest cells again. They migrate all over the embryo to form pigment cells, peripheral neurons, facial bones, and more. A key survival factor for many of them is stem cell factor, SCF, which binds to its receptor, KIT, on the neural crest cells.
What happens if that fails?
If there are mutations affecting SCF or KIT, many migrating neural crest cells undergo apoptosis, programmed cell death, because they don't get the survival signal. This can lead to defects like patches of unpigmented skin or fur, piebaldism, because the pigment cell precursors didn't survive to populate those areas.
The cells don't always travel alone, though. Sometimes they move in groups.
Right. Many important morphogenetic movements involve collective cell migration, where cells migrate as coordinated groups, maintaining cell-cell contacts and exhibiting collective behaviors.
What kind of groups?
It takes several forms. There's chain migration, where cells follow each other in stream-like files. We see this with some populations of neural crest cells navigating through the embryo. Then there's clustered migration, where cohesive groups move together, often with specialized leader cells at the front that seem to guide the follower cells. The migration of the zebrafish lateral line primordium, which forms a sensory organ along the fish's body, is a beautiful example of this.
Like a little convoy.
Exactly. And finally, there's sheet migration, where entire epithelial sheets move coherently across a surface. This is crucial in processes like wound healing, where epithelial cells at the edge of a wound migrate collectively to cover the gap, and also in development, for instance, during the fusion of the palate shells in the roof of the mouth.
Determining size.
Okay, Fani, let's tackle one of the most fundamental yet still quite mysterious aspects of development: how the size of an animal or an organ is determined. Why are we so much larger than a mouse? Or, why is a Great Dane so different in size from a Chihuahua? It's not just about shape, it's about scale.
Right. Size control is a huge question. Fundamentally, the size of an organism or organ is defined by three key variables. First, the total number of cells. Second, the average size of those individual cells. And third, the quantity of extracellular material, the matrix surrounding those cells.
So, more cells, bigger cells, or more stuff between cells?
Basically, yes. For instance, humans are vastly larger than mice, primarily because we have vastly more cells. Differences between wild and cultivated plants, or different breeds of dogs, can involve differences in both cell number and cell size. The deep mystery that fascinates scientists is how cell proliferation, cell growth, increase in size, and programmed cell death, apoptosis, are so precisely regulated and coordinated across the whole organism to produce that characteristic final size for a species or an organ.
It seems like there must be some kind of measurement going on. How does an organ know when it's big enough? Those classic transplantation experiments hint at this, right?
They do. If you transplant multiple fetal thymus glands into a developing mouse, each thymus tends to grow to its normal individual adult size. This suggests some kind of intrinsic local size control mechanism within the organ itself.
Okay, self-contained growth.
But if you do the same with fetal spleens, transplant several into one host, each individual spleen ends up smaller than normal, but collectively they add up to the mass of about one normal spleen.
Whoa. So the body somehow knows the total amount of spleen.
Yeah. It implies some kind of systemic feedback mechanism, perhaps involving circulating factors, that assesses the total functional mass of certain organs like the spleen or liver and adjusts growth accordingly. It's like the organs can somehow sense their total size within the context of the whole organism. The tiny worm C. elegans provides a really clear, albeit simple, illustration of how proliferation, death, and cell size contribute to organism size. Remember, it's precise cell lineage.
Yeah, exactly. 959 somatic cells in the hermaphrodite.
Right. That precise number is achieved through a perfectly programmed schedule of cell divisions and a significant amount of programmed cell death removing unwanted cells. So, cell number is fixed. But what's fascinating is that after the cell divisions stop, the worm continues to grow substantially, often doubling in size.
Bigger cells.
Bigger cells, exactly. This post-mitotic growth happens largely through endoreplication. The cells undergo rounds of DNA synthesis, duplicating their chromosomes, but without actually going through mitosis or cell division.
So they become polyploid, multiple chromosome sets.
Precisely. They become polyploid, and this generally leads to a dramatic increase in individual cell size. We see extreme examples in other organisms too, like giant neurons in the sea slug Aplysia that can become massively polyploid.
This link between ploidy and cell size seems quite general then.
It often is. You mentioned the frogs. The tetraploid Xenopus laevis is about twice the size of its diploid relative Xenopus tropicalis, primarily because its cells are larger. In agriculture, intentionally creating polyploid plants is a common strategy to get larger fruits, flowers, or vegetables.
But then you have those weird salamanders.
Ah, yes, the counterintuitive example. Some salamander species have huge genomes and correspondingly huge cells due to polyploidy, but instead of becoming giant salamanders, they compensate by having fewer cells overall.
So bigger cells but fewer of them means the organ stays the same size.
Roughly, yes. It maintains the overall organ and organism size. This strongly suggests that organisms, or at least certain organs, have mechanisms to assess total cell mass or volume, not just cell number. It's like they have a biological scale for tissue bulk. So these increases in cell size often result from specific cell cycle modifications like endoreplication, DNA synthesis without mitosis, or endomitosis, nuclear division without cell division, leading to multinucleated cells. We see this in specialized cells like the giant trophoblast cells of the mammalian placenta, which are crucial for nutrient exchange.
But how genome size correlates with cell size? That's still fuzzy.
The precise molecular mechanism linking the amount of DNA to the size of the cell is still one of biology's major outstanding mysteries. We know it happens, but the why and how are not fully understood.
The idea that animals and organs can actually assess and regulate total cell mass is just incredible. You mentioned the Drosophila imaginal discs.
Right, the imaginal discs, those little sacs of cells in the fly larva that will develop into adult structures like wings, legs, and eyes, provide a striking example of this homeostatic size control.
How so?
You can experimentally manipulate cell division rates in a developing wing disc. You can speed it up or slow it down significantly, but remarkably, the final adult wing that develops is still almost perfectly normal in size and patterning.
So it compensates. If cells divide slower, they divide for longer.
Exactly, or if they divide faster, they stop earlier. It indicates there are intrinsic regulatory mechanisms within the disc that monitor its size and halt growth and proliferation when a specific target size is reached, regardless of the cell number or the time taken to get there. It's like an internal stop growing now signal is triggered at the right size.
How does that signal work? Are specific molecules involved?
We're still unraveling the details, but we are identifying key extracellular signals that stimulate or inhibit growth. In that Drosophila wing disc, for example, the same signaling protein DP, related to vertebrate BMPs, that helps pattern the wing also influences its overall size. Gradients of DPP seem to provide both positional information and growth-promoting cues. But growth isn't just about go signals, there are also crucial stop signals. Myostatin is a fantastic example. It's another member of the TGF family, like DPP BMPs, but it specifically acts to inhibit muscle growth and proliferation.
So it puts the brakes on muscle size.
Exactly. And when the myostatin gene is deleted or mutated, the brakes are off. Muscles can grow several times larger than normal. This is seen dramatically in certain cattle breeds selected for heavy muscling. And in those bully whippet dogs that accidentally acquired a myostatin mutation, it's a very clear negative regulator of organ size.
And then there's the hippo pathway. That sounds important.
It's hugely important. It's an intracellular signaling pathway, first discovered in Drosophila, but highly conserved in vertebrates, that acts as a major brake on tissue growth.
Why hippo?
It was named after the hippopotamus-like overgrown tissues, huge heads, massive eyes that result when the pathway is inactivated in flies. Normally, the hippo pathway functions to inhibit growth by promoting apoptosis and inhibiting cell cycle progression, primarily by keeping a key transcriptional co-activator called Yorkie, or YATTZ in vertebrates, out of the nucleus.
So when hippo is off, Yorkie goes wild.
Exactly. Yorkie enters the nucleus and turns on genes that drive cell proliferation and block apoptosis, leading to dramatic tissue overgrowth. What's particularly fascinating about the hippo pathway is that it seems to be regulated by a variety of inputs, including mechanical cues like cell density, cell shape, and the stiffness of the extracellular matrix. Cells literally sense their physical context to control their growth via this pathway.
Okay, so we have local signals, internal pathways.
Yeah.
What about body-wide coordination? Hormones must play a role.
Absolutely. Hormones provide systemic body-wide coordination for growth, especially in vertebrates. The most famous example is growth hormone, GH, secreted by the pituitary gland.
Gigantism and dwarfism.
Right. Too much GH during development leads to pituitary gigantism, and too little leads to pituitary dwarfism, where individuals are proportionately small but otherwise normally formed.
But not all dwarfism is like that.
Correct. Achondroplastic dwarfism, the most common form, results in disproportionately short limbs compared to the trunk. This is usually caused by a specific mutation in a receptor for another growth factor, FGF, fibroblast growth factor, which primarily affects cartilage growth in the long bones. GH itself mostly acts indirectly. It stimulates the liver and other tissues to produce another factor called insulin-like growth factor 1, IGF-1. It's largely IGF-1 that then circulates and promotes cell survival, cell growth, size increase, and cell proliferation throughout the body. And of course, overall nutrition status profoundly influences these hormonal networks and growth.
So rate of growth is controlled. What about the duration of growth? Why do we stop growing while some animals seem to keep going?
That's another key aspect of size control. We distinguish between two main patterns. Determinate growth is typical for species like humans, birds, and many insects. We have a defined period of rapid growth, usually ending around sexual maturity or metamorphosis in insects, after which growth largely ceases.
We reach a final size.
We reach a characteristic final size, yes. But other species exhibit indeterminate growth. Think of many fish, reptiles, amphibians, lobsters, and essentially all plants with their continuously active meristems. These organisms are capable of continuous growth throughout their life as long as environmental conditions are favorable. They don't have a rigidly predetermined final size.
And do we know what controls the difference? Why determinate versus indeterminate?
Honestly, the precise molecular mechanisms that switch growth off in determinate growers or allow it to continue in indeterminate ones are still largely unknown. It's likely tied to the complex interplay of genetic programs, hormonal changes associated with maturity, and possibly limitations imposed by scaling and physiology, but it remains a major area of ongoing research.
Wow. That was truly a deep dive into the incredible world of animal development, wasn't it? From a single cell orchestrating its destiny to a complex organism. It's just an amazing story of conserved mechanisms, intricate signaling, precise timing, and that dynamic cellular choreography that literally builds us from the ground up.
It truly is. And we've seen how a surprisingly small set of molecular tools, those highly conserved signaling pathways like WANT, hedgehog, notch, and those master regulatory genes like HOX or PAX6 can be used to build such a vast diversity of life.
It's like evolution is the ultimate tinkerer.
Absolutely. It's a real testament to evolution's ingenuity, constantly reusing and remodulating these basic building blocks. And often, as we discuss, the big changes come not from inventing new parts, but just by changing the instruction manual, the regulatory DNA, to create these endless, spectacular forms.
And perhaps the biggest takeaway for me is how deeply connected all these processes are. I mean, from the initial patterning of an embryo to the growth and maintenance of our adult bodies, and even how we respond to our environment throughout life.
Mm-hmm.
These aren't just, biological curiosities tucked away in embryology textbooks. They really inform our understanding of health, how birth defects arise, how our tissues repair themselves, and even how diseases like cancer develop when these very processes go wrong.
It definitely makes you wonder, doesn't it? Given the incredible precision and interconnectedness of these systems, what subtle shifts in these fundamental rules, maybe just a tweak in regulatory DNA, might have led to some of the most striking evolutionary innovations we see around us? A wing, a bigger brain, losing limbs.
Yeah.
And what might still be uncovered about those long-term chromatin timers we touched on? The mechanisms that seem to govern our developmental trajectory, maybe even aspects of aging over years or decades. There's still so much to learn.
Well, we hope this deep dive has given you a newfound appreciation for the intricate cellular dance that makes us who we are, and maybe sparked some curiosity about the profound questions that still challenge scientists in this field. Keep exploring, keep questioning, and join us next time for another deep dive. Thanks for being a part of our last-minute lecture family.